This is the screencast for cellular injury, the lecture number 2. So in this lecture we are going to discuss certain learning outcomes. So in the end of the lecture, the student can able to define cellular injury and explain the types, list the causes of cellular injury, and they can able to explain the pathogenesis of cellular injury in various clinical settings such as ischemia, free radical, and radiation. And they can able to compare and differentiate the morphology of different types of necrosis, and they can able to define the apoptosis and the causes of apoptosis and various mechanism involved in apoptosis, and they can understand the clinical importance of apoptosis. Okay, so before going into the cellular injury, let us revise the basics. And you all know about the cell, and cell has cell membrane, and the cell has nucleus. Inside the nucleus we have chromatin, reticulum, chromatin, reticulum, and uh, then we have tubular structure which is studded with ribosome, and there are 4 ribosomes available, and this is called rough endoplasmic reticulum, and this is ribosomes which contains RNA, and the chromosome reticulum contains DNA, and we have Golgi apparatus, and these 3 are involved in protein synthesis. And you have smooth endoplasmic reticulum, and we have mitochondria. We have mitochondria, and this is smooth endoplasmic reticulum, okay, which stores the calcium, and even mitochondria stores the calcium. And apart from that we have lysosomes, lysosomes which contains enzymes, okay. So the mitochondria produces energy by using oxygen and produce ATP, and this is called oxidative phosphorylation, and otherwise called as aerobic respiration. And we have outside the cell, in the cell membrane we have sodium potassium ATPase channels and also other channels, sodium potassium ATPase channel, and outside the cell it is called extracellular environment, it is called extracellular environment. In extracellular, sodium is more, potassium is less, sodium is more, potassium less. Intracellular, sodium is less, potassium is more, and this is maintained by the help of sodium potassium ATPase channel which needs ATP. So this ATP comes from the mitochondria, so that the cell survives. So for the production of ATP, you all know we need oxygen. If there is no oxygen, they use glycogen to produce ATP, and if the glycogen is used, they will produce more lactic acid. This lactic acid is acidic one, so it will decrease the pH. So when the cell receives good blood supply from the blood vessels. They have RBCs. This is a blood vessel which has RBCs. RBCs has hemoglobin. Hemoglobin contains oxygen, and the oxygen freely diffuse to the cell, and it goes to the mitochondria. It goes to the mitochondria. In the mitochondria, the ATP is produced. This ATP from the mitochondria, it goes to the sodium potassium ATPase channel, and ATPase is the enzyme. ATPase is the enzyme which breaks the ATP and they produce energy, and the sodium potassium channel is maintained. Okay, so if there is any block, what happens? This blood supply is cut. So the loss of blood supply, loss of blood supply, this loss of blood supply is called ischemia. So ischemia will cause oxygen decrease. Okay, so oxygen decrease. This oxygen decrease is called hypoxia. If there is hypoxia, then ATP is reduced. So if there is hypoxia, what happens? If there is ischemia that produce hypoxia and ATP is cut down, then they use glycogen, and the glycogen will produce lactic acid, and they will generate ATP. This ATP goes to the sodium potassium ATPase channel. So what happens is, once the glycogen is over, then this ATP also cut, then pH is reduced because of the lactic acid. So now the sodium potassium channel is not getting any ATP, so it's not getting any ATP. Then the sodium potassium channel is opened up. So because of more concentration in the sodium outside, so all the sodium enters into the cell, because sodium potassium channel opens up. So sodium potassium channel opens up, all the sodium enters into the cell. Now the sodium content is increased. Whenever the sodium comes, it comes with water. So water is also increased. So now more fluid is coming. Inside the cell, and other channels like calcium channel is also opened up, and calcium is also coming more. When calcium comes, they try to go inside the smooth endoplasmic reticulum. So what happens? The calcium is already full in the smooth endoplasmic reticulum. They cannot enter. The calcium is increased in the cytoplasm. Okay. So now the cell is, what happens to the cell? The cell literally swells. So the nucleus is also swelling, mitochondria is also swelling, and endoplasmic reticulum also swelling, Golgi apparatus also swelling, lysosomes also swelling. Nucleus, mitochondria, and ribosomes comes out of the endoplasmic reticulum, and Golgi apparatus all are swelling, and smooth endoplasmic reticulum also swelling. And mitochondria is also swelling. So all are swelling, the cell is swelling. In this stage, if you give back the oxygen, the cell comes back to normal. The cell comes back to normal. The cell comes back to normal. So once again I am telling, when there is no oxygen, once there is no oxygen, then the ATP is lost, then they use glycogen and they produce more lactic acid, the pH is reduced. Once the glycogen is over, then that ATP is also lost. That will open up the sodium potassium ATPase channel, that will open up the sodium potassium ATPase channel, and more sodium is coming, followed by the water. Then calcium channel gets opened up and more calcium is increased, and that all will cause more pH reduction. More pH is reduced and cell is getting more fluid and as well as electrolytes, cations inside, so the cell swells. So when you give oxygen at this stage, the cell will come back to normal. So this is the stage that is reversible. Understand? If you oxygen is not given at this stage, then the cell cannot come back to normal, and the cell, what happens is the lysosome ruptures. The lysosomes rupture, the lysosomal enzymes comes out, and you all know the lysosomal enzyme which is coming out gets activated because of calcium, because we know the calcium is now more inside, and the mitochondria also sends all the calcium out, and smooth endoplasmic reticulum also send all the calcium out, and this lysosomal enzymes gets activated, and these lysosomal enzymes are powerful and they can able to break the DNA, they can able to damage the DNA, this lysosomal enzyme, or they can able to damage the all the proteins in the cell by the enzyme protease, and they can able to damage all the phospholipid by enzyme phospholipase, and they can also damage the remaining small ATPs in the cell by ATPase. So they have DNase and protease, phospholipase, and ATPase. This all the enzymes get activated. Because of calcium. Understand? So these enzymes will completely break the cell and destroy the structures of the cell, and this is end up with a cell death. This is called necrosis. This is called necrosis. This necrosis will induce inflammation. 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 So necrosis will induce inflammation. So this all happens because of no oxygen. So this all happens because of reduced oxygen, hypoxia, and this happens because of ischemia. Okay, so ischemia produce hypoxia, and this hypoxia will result in necrosis. So this is the mechanism of necrosis. So just I am removing this. Okay, now just I will summarize the things. You all know this is a cell, and cell has nucleus, and you all know nucleus, they have mitochondria, and they have smooth endoplasmic reticulum, and they have rough endoplasmic reticulum, right? And they have Golgi apparatus and lysosomal enzymes, and this all will be good because if they get oxygen. If there is no oxygen, what happens? The mitochondria cannot produce ATP. Then the ATP, then they use glycogen to produce ATP, and that will increase the lactic acid. Then the ATP, then once the glycogen is over, the ATP is over, and the sodium potassium channel opens up, and more sodium, more water comes along with calcium. So the calcium also escapes out from the mitochondria and the endoplasmic reticulum. So the cell will swell. If you give oxygen here, that is reversible. That is the stage of reversibility. If you are not giving oxygen at this stage, then the cell will rupture. The cell will rupture, and lysosomal enzymes also rupture. The enzymes leaks out. The enzymes leaks out, and they will completely destroy the cell. They will completely destroy the cell. The enzyme gets activated because of calcium. So this is what happens in the ischemia. Ischemia means loss of blood supply, which produce hypoxia, then that end up with necrosis. Okay, so now I am going to describe a different concept. During the process of oxidative phosphorylation, that is oxygen is producing ATP, that time some chemical substances are produced, that is called free radical. Which means the chemical substance at the outer orbit, they have single electron, unpaired electron, so there is empty electron there. So they try to get electron from all the chemical substance, from cell membrane, from nucleus, from any protein, any cytoskeleton. So whenever they see, they take electron. So during normal oxidative phosphorylation, this free radical are produced. This free radical in oxidative phosphorylation, they produce the substance called reactive oxygen species. Otherwise called as ROS. So when the reactive oxygen species are produced, that has to be balanced by a antioxidant. This antioxidant donate electron. So example of one antioxidant, glutathione. Okay, they have lot of electron, they donate electron, and they will balance it. But in smoking, these free radicals are increased, these antioxidants are decreased. So the reactive oxygen species is increased, so that is called oxidative stress. That is called oxidative stress. So this oxidative stress can damage the nucleus, directly. When the cell nucleus is damaged directly, that will end up with a cell death called apoptosis. So the cell will become into multiple pieces. The cell will divide into multiple species, and that will be removed by the macrophage. So they will not induce inflammation. There is no inflammation. So this is the characteristic feature of apoptosis. So any direct damage to the nucleus, for example, oxidative stress. Other examples are viruses, radiation. So any damage directly happening to the nucleus, okay, that will end up with apoptosis. So this is a pattern of cell death. So now let's go to the slides. So if the adaptive capacity of is exceeded, cell injury develops. So cell injury may be reversible, or if the cell can return back to normal. If the stress is persistent, the cell goes for irreversible cell injury, results in cell death. So reversible cell injury means if the injury has typically not progressed to severe membrane damage and nuclear damage. So that is the stage called reversible injury. So it should not progress to membrane damage. If it is continuous and the injury becomes irreversible because membrane is damaged, so the cell goes for cell death. There are two types of cell death: necrosis and apoptosis. So there is oxygen deprivation is one of the most commonest cause for cell death, that is called hypoxia. So one of the most commonest cause for hypoxia is ischemia, which means loss of blood supply. So there are some chemical agents which can directly cause damage to the cell membrane that can lead to cell injury and cell death. So some cells they die immediately, some cells takes 3 hours to die. Example, skeletal muscle cells. Some cells die after 30 to 40 minutes. Example, cardiac muscle cells, because of the storage capacity. So what are the components of cell are affected? Mitochondria is affected when there is hypoxia, and cell membranes are affected, and protein synthesis are affected because endoplasmic reticulum, Golgi apparatus and ribosomes are affected. Cytoskeleton is affected because of cell is swelling, and genetic apparatus of the cell is affected. The DNA is affected in the end. So now if there is ischemia which causes decrease in oxidative phosphorylation, which will reduce the ATP formation, and then there is a anaerobic glycolysis, that is they use the glycogen and they in the end they produce lactic acid that will decrease the pH, and then the sodium potassium channels cannot work, and then there is influx of more calcium, hydrogen and sodium. Then all the cellular organelles goes for swelling, and there is a detachment of ribosomes that end up with decreased protein synthesis. So calcium enters the cell more, and the calcium also comes out of the mitochondria and endoplasmic reticulum, and they will activate these 4 enzymes. So ATPase and phospholipase, protease, endonuclease, which is coming from the lysosomes, and then they will degrade the ATP. ATPase will degrade the ATP, phospholipase will degrade the phospholipid, and protease will degrade the cytoskeleton, and endonuclease will degrade the nucleus. Then completely end up with severe membrane damage, and they will go for necrosis. So calcium activates the enzymes, and ischemia and toxin cause increased cytoplasmic calcium, which is very dangerous. Okay, sometimes the direct damage to the DNA, example radiation injury or oxidative stress, can initiate the cell to go for suicide, that is called apoptosis. The type of cell death is called apoptosis. So when the cell is swelling, then if you give back the oxygen or you remove the cause of injury, then the cell becomes reversible. If the cell continues with that injury, then it results in rupture that will lead to irreversible injury, ends up in necrosis. Remember, all pathological form of cell death, pathological form of cell death is necrosis. So now coming to the free radicals. So free radicals are the substance which has unpaired electron at its outer orbit. Antioxidants are the substance which donate electron. If free radical are increased, then they will take all the electron from the surrounding substance and they make them into free radical, and they will completely damage the cell. So free radical are chemical species with a single unpaired electron in an outer orbit. So they are extremely unstable substance. They always react with all chemicals, inorganic or organic chemicals, and they will convert them into free radicals. So it is a chain reaction. So example, oxygen derived free radical, that is reactive oxygen species, which is produced normally in cells during mitochondrial respiration and during energy production, energy generation. So they are degraded. This naturally produced reactive oxygen species are degraded by a scavenging system, that is antioxidant system called glutathione. Example, glutathione. So if the production of reactive oxygen species increases, or the scavenging system, that is antioxidant mechanism, is ineffective, that results in excessive reactive oxygen species, that is called oxidative stress. That is called oxidative stress. So example, radiation injury. In radiation injury, oxidative stress is formed. So now coming to the types of necrosis. Coagulative necrosis. This is a morphological appearance of necrosis, and it is seen in all the organs except brain. In the brain you will be seeing liquefactive necrosis, and hypoxic death of cells in central nervous system, that is brain, it produces liquefactive necrosis. And infection also change the coagulative necrosis into liquefactive necrosis. Understand? Infection can change coagulative necrosis into liquefactive necrosis. If the blood supply to the leg or the limbs is lost, then the feet will go for coagulative necrosis, that is called dry gangrene. Dry gangrene. If it is infected, then it will become liquefactive necrosis, it is called wet gangrene. It is called wet gangrene. If it is infected by clostridium, it is called, that will produce gas, that bacteria will produce gas, it is called gas gangrene. It is called gas gangrene. So fat necrosis means in an alcoholic person, the pancreatic lipase, okay, is increased, that will destroy the fat, and it is seen in a condition called acute pancreatitis, acute inflammation of the pancreas. So you can see a white area, chalk like white areas can be seen. Now coming to the apoptosis. So apoptosis is a form of cell death due to enzymes destroy its own DNA. So the DNA is damaged first, and it is called programmed cell death. So it can be physiological. Example, in embryogenesis, when the embryo is developing, some part of the tissues go for cell death by means of apoptosis. Pathological can be seen in radiation injury, which causes direct damage to the DNA, and even viruses and oxidative stress that will produce apoptosis. Now coming to the difference between necrosis and apoptosis. Necrosis, there is membrane damage, and the lysosomal enzymes digest the cell, and it triggers inflammation, and it is always pathological. Apoptosis, it is direct damage to the nucleus. Dissolution means nucleus becoming like a fluid, complete dissolution of the nucleus, and this is due to the activation of the enzyme caspases, whereas in necrosis it is lysosomal enzymes. Apoptosis may be physiological and pathological. Common pathological cause are virus and radiation injury. This ends the lecture number 2.